Part of next week we'll be talking about microscopes. As our introduction, we need to know how to use a microscope, what the various parts are, so that we can move ahead and when we get into the cells portion, so we can actually look at some cells under the microscope. So we'll just introduce microscopes first. Size the whole unit. You can refer back to it as we move through the unit. But we're going to talk about 3 types of microscopes, and we will actually use 2 types of microscopes. So the first type of microscope is the one we'll use most frequently. This is the version of the microscope you will use. This is a little bit larger version. This is one that can hook up to the computer. But they're basically the same, and they work in the same way. These are called the compound light microscopes. Compound light microscopes. They're called compound. You know what compound means?
Like a compound is 2 elements combined.
It is. The word itself, like a compound word, 2 words combined. Compound just means sort of more than one part. And a compound microscope is a microscope when the image gets magnified by 2 separate lenses. The other type of microscope we'll use in class is this kind of microscope over here. This is referred to sometimes as a stereoscopic microscope or a dissecting microscope. The compound microscope gives you a magnification of about 40 at the low end to up to about 400 times for our microscopes. Some compound microscopes can get up to about 1,000 or so. But our microscopes go from 40 to 400. The dissecting microscope has a much lower rate of magnification. It can magnify from about 10 to 30 or 40. It depends on the microscope you got. And then there's one other type of microscope. Shown here, we don't have one of these. I mean, they cost $100,000 or so. You may find them in a research lab or a university or something. This is called the electron microscope. And the electron microscope, rather than making an image using light, it uses a very, very tiny beam of electrons, which gives it excellent resolution and gives it a great ability to magnify an image. The electron microscope can magnify an image up to 1 million or sometimes even 2 million times. So it's very, very powerful.
Besides the magnification rate, like what is the difference between the compound light and the dissecting?
We'll talk about it in 1 minute, okay? Because there are some important differences. All right, so those are 3 types of microscopes. If we look at some images from the compound microscope, here are some single-celled living things. You're going to see almost all of these. You're going to see amoeba later on. This is paramecium. This is euglena. This is vorticella, foraminiferan, radiolarin. So these are all living creatures, single-celled living creatures, that are very small, and we can use the microscope to see them. Now, what you'll notice when you're using the compound microscope, the image you see is a very flat, 2-dimensional view. In fact, the way the microscope works is light from down here has to go up. It goes through a hole in this thing, which is the stage, and then it has to go through the object and up through the lenses to your eye. So could I look at my hand under here? No, because light is not able to pass through my hand. I'm not going to see anything. Objects that you're looking at in the compound microscope must be very, very thin. Okay? So it only really gives you a 2-dimensional view of your object, as we see here. You know, if you want to look at some muscle, what you have to do is you basically have to make thin slices of the muscle, very, very thin, so light can pass through it. You can't look at solid objects in the compound microscope. But in the dissecting microscope, if you imagine, The light works a little differently. Now, could I look at my hand in this microscope?
Yes.
Yes. Now the light here is coming off of the top, bouncing off my hand, and going through the lenses. I would be able to look at my hand. I could look at a solid three-dimensional object. The other important difference about the dissecting microscope are the two eyepieces. Two eyepieces allow you to get a good sense of the depth of the object. Do you know what depth perception is? You know what that? Have you heard the term at least?
Yeah.
Yeah, it's sensing how near or how far something is. Humans have very good depth perception. Do you know why? Luke?
Because we have two eyes.
Yeah, but so does like a fish, but fish have poor depth perception. Think about the difference in the eyes. You're on the right track. Joey?
Like a fish's eyes are on the side, but humans are right in the
Yeah, two forward-facing eyes are generally adaptations of animals that need a good sense of depth perception, like a cat or a raccoon or a chimpanzee. And the reason that helps with depth perception, I could illustrate it. If you ever, I'm sure you've done this before, if you hold like your finger up close to your face, and then you open and close one eye and the other, right? Has everyone done this before?
Yeah. No.
What happens?
It moves.
Yes, it appears to move. Why does it appear that my finger is moving when I do this? Courtney?
Your eyes are on different sides.
Yeah, because my eyes are on two sides of my face, each eye is seeing my finger at a different angle. And so it looks like it's in a different place. Okay? Now if you stretch your hand out farther and do the same, it still moves. But how much of a difference is there? Less. If you look across the room at an object, how much difference is there? Very little. What happens is our eyes are always seeing sort of two different versions of the world because they're each looking at a slightly different angle. But our brain takes these images. Anything that our eyes see at a very different spot, our brain says that must be close to you, just like your finger here. Each eye is seeing it at a very different spot. Your brain tells you, well, this is very close to you. Objects that look basically the same from both eyes, our brain tells us that must be very far away. And so having two eyes allows us to get a good sense of depth perception. You know, if you ever, got something in your eye, or you lost the contact, or you couldn't see very well, and tried to like play sports or catch something, it's much harder if you can only see out of one eye, because you lose a lot of that sense of depth. It's also how three-dimensional movies work. You have to put on those glasses. What those glasses do is they allow each of your eyes to see a slightly different version of the movie. And the people that make the movie, if they want the object to look like it's right close to your face, okay, they make it very different. Okay? Or did you ever see the like blue and red drawings with the glasses? Same idea. They draw the image in red and in blue. The parts of the image they want to look like they're very close to you, they draw the two images separate. The parts they want to look very far away, they're right next to each other. So two forward-facing eyes allows us to have a sense of depth. And the dissecting microscope allows us to see sort of three-dimensional view of an object, like this aphid. This is a planaria, a flatworm. This is a seed that's starting to germinate. And so you can see the depth much better using the dissecting microscope. All right, and finally the electron microscope. The electron microscope, again, it can give great resolution, great magnification. Here are some things viewed through the electron microscope. There's a spider, some toilet paper. This is Velcro, human hair. Now these are artificially colored. Generally, the electron microscope only gives a black and white image. Sometimes they're artificially colored just to make things sort of stand out a little better. This is a fly. You can see its eyes, but on top of the fly, these are mites, parasitic insects that live on it. That's another example. That's another mite living on a beetle.
Is that actual size? Or is that like?
No, those are all large. Maggot. Some more insects. Yeah, those are its mouth parts. They also have two little hands. Alright. So, what we need to do is talk about parts of the compound microscope. You're going to have to know these parts. You'll have a quiz on it next week. So, it's important that we understand what the parts do, how to use the microscope in order for us to view objects appropriately. So, we'll just go through these. Our microscope that we generally use is this one. Looks a little different from this one, but I'll point out those differences. First thing is where the lenses of the microscope are located. So when we're looking through this microscope, where are the lenses? Let's see. Jonathan?
Me? Oh, in the eye.
Yeah, there's one up here.
And then there's one down. Right.
And then there are lenses here. This one's called the eyepiece. These are called the objective lenses. Hello? Oh, okay. The part where these objective lenses attached to, okay, that's called the nose piece. You rotate it to select the various lenses. An important thing to note is that as you rotate the nose piece, it will click in. You have to be clicked into one of the notches in order for the microscope to work. It can't be in between. The body tube. The body tube is this long tube that sort of separates the objective lens from the eyepiece. Light has to travel through that. And then on the bottom we have the light. So here is the light on this microscope. On this microscope, the light's down here as well. Okay. Our microscopes that we generally use are cordless. They have a rechargeable battery and an LED light, so you could bring them to your desk. Some need to be plugged in, like this one. This one, however, doesn't have an actual light. What does it have? Nicole? Yeah, that's a mirror that reflects light. That's sort of the older way microscopes used to work, is you'd have a separate light that you'd sort of aim at that mirror, and then you'd adjust the mirror so that light reflects up through the object. We don't use those anymore, but that should be the light. Then this flat part where you actually put your specimen, it's called the stage. And when you're using the compound microscope, you're almost always looking at something on a slide, a small piece of glass. Okay? And so you need a way of holding it down. Some microscopes like this one have these stage clips. You see? It's like these. It's just a little clip that you put the slide under those metal clips. But these microscopes are a little different. The one you use has what's called a mechanical stage, where you have this like spring-loaded clip that holds your slide in place. But then you just move the slide by turning these knobs. That moves this whole mechanical stage forward and backward. It allows you to move things much more accurately than trying to move it around with your fingers. So our microscopes that we're generally going to use work a little bit differently. Underneath the stage, there is a thing called the diaphragm. And on some microscopes, it's this dial. And this dial has different holes in the bottom. And by changing which setting you're on, you can make your image brighter by using one of the larger holes so more light goes through, or a smaller hole would allow less light, making it not as bright. So that part is called the diaphragm. Then we have the parts that sort of support the microscope. On the bottom is the base. This part here is called the arm. And then we have two important adjustments here. Now in this microscope you'll see there's this knob here, and what you can see what it does, it moves the stage. Now sometimes people think this zooms in on the object, but it doesn't. You know what it does? Ashley? Yeah, it focuses the image. Okay? It focuses the image so that it's clear. The big knob is called the coarse adjustment. It moves the focus a lot. Then there's a smaller knob which also moves it, but it moves it a very small amount at a time. That's called the fine adjustment. And that's if you want to fine tune it when you're under high power, you would use the fine adjustment, because it only moves it a tiny amount. Now our microscopes are a little different. Our microscope has just one knob here. We have this larger knob. That's the coarse adjustment on these microscopes. And then the smaller part here, that's the fine adjustment. So those are the parts of the microscope. So again, if we go through, we have the eyepiece, the body tube, the coarse adjustment, fine adjustment. We have the nose piece and the objective lenses. We have the arm, stage clips, diaphragm, and the light, and the base. Okay? We don't have a condenser control knob on ours. All right, last thing, let's look at a few more pictures. These are kind of cool. Tell me if you can guess what we're looking at. So under the electron microscope. Any guesses, Joey? No? Emma?
Is it a fork?
No, it's actually a CD.
Oh.
It's the layers, the metallic layers that make up a CD or DVD.
It seems like it's really zoomed out.
It's not. Morgan?
Is it like a razor?
Yeah. These are the blades of a razor,
Oh, that's
and these are little hairs that just got shaved off of somebody.
Oh, yeah.
Little flakes of skin.
I thought that was like shells.
No? It's magnified, so it's,
Yeah, I thought it was the side of the street.
it's not s'more cereal, but it is something edible. It maybe even is something you ate this morning, especially if you had eggs or something. Yep, salt and pepper.
Really? That's salt and pepper?
That's like marshmallows and chocolate.
I know.
What?
Jelly. Jelly.
Jello,
No?
Is it edible?
No. You may have this on your jacket in your locker.
Zipper? Is it Velcro?
Velcro. Velcro works by, you know the rough side of the Velcro? That's a bunch of these plastic little hooks. You see the hooks. The softer side is this sort of weave of loops of fabric. When you push them together, the hooks kind of grasp on to the loops, and it stays together. When you pull it apart, these hooks are kind of flexible. So as you pull it apart, those hooks flex, the loops detach, and then you can separate.
So why when it's white? Why is it red?
Just the color it was.
Pasta.
Not pasta.
Garlic.
No.
Paper.
Toilet paper. This one I don't know if anybody would ever get, but no. No, but you're getting closer.
What'd she say?
Rope, not string.
What is cable?
Fishing wire.
No. Rubber band. You're getting closer. It's not quite rubber, but it's an elastic material.
Rubber band.
No. This is what, a nylon stocking looks like. So stockings that a girl would wear, they're made of these strands of this flexible material called nylon, which can flex, and it's all woven together. All right.